Okay, so good morning, everyone. Um, so let's continue our discussion about the role that our immune system plays in fighting cancer, and importantly, um, how cancer cells, in the process of becoming cancer, have to learn to evade or work around the immune system, right? So that the reality is if you even end up with a large tumor representing a cancerous lesion, then by definition, those cancer cells must have at some point stumbled upon a series of mutations that enabled them to get around the body's immune system, detecting and destroying them. Last time we talked about how important it is to think about that cells in our body are constantly advertising or presenting evidence of what's happening inside of them in order for our immune system to have the opportunity to check them out. Right? I said last time that cellular components of our immune system, things like the macrophages, or dendritic cells, or T cells, natural killer cells, right? These different types of white blood cells or leukocytes, they're very good at detecting things that are, outside of cells, in between cellular spaces, especially things like macrophages. Right? They can easily go around between cells, finding things to eat up and destroy, like bacteria. Right? More developed lymphocyte cells, like our T cells and B cells, right, they can only come in contact with things like antigens, well, B cells floating around in your tissues between your cells. They can encounter antigen there, right? But they don't really have an opportunity to peer inside of cells of your body to see what's going on inside of them. Right? I kind of joke about that. It's not like a cell, like an immune cell can come up to another cell and actually take a peek inside of it to see what's going on. Right? That's why it's up to the cells of your body to constantly be presenting on their surface evidence of what's happening inside of them. And they do so using the major histocompatibility protein complexes, or MHCs. And we said last time, as a review of immunology, there are two main types of these MHC platforms for presenting things. There's MHC class 1 and MHC class 2. And depending upon which platform a particular small peptide is being presented on, right, that will have a tremendous impact on how the immune system sees it, right, what immune cells are able to detect it, and the repercussions or the consequences of the cell that's doing the presenting. So in this cartoon here, we're looking at two types of immune cells, dendritic cells and B cells. In both of these cases, the cells, so the big yellow things, right? The big yellow things are the cells. On the top one, you can see dendritic cells are constantly taking in different types of bits and pieces of things around them. So in this case, maybe these are 3 different proteins that are being taken in from the surrounding extracellular matrix, or the fluid, for example, between the tissues or within the tissues. Inside the dendritic cell, these proteins are processed, right? They're broken down into short little oligopeptides, peptides that are maybe on the order of 10 to 20 amino acids in size. Right? Inside of this dendritic cell, you also have these MHC class 2 protein platforms, these yellow structures here, that were made in the endoplasmic reticulum and then budded off inside of a vesicle. And these vesicles are then going through the cytoplasm, and they're fusing with the contents of the endosome, right, these little peptides, to form what we call the late endosome. And in this situation, these different peptides are now being loaded on MHC2 platforms, right, you can see here, and then they are brought to the surface of the cell to present. So in this case, the dendritic cell, it doesn't really know what it is encountering and taking in and processing, right? Its job is just to go around and collect as much stuff as it can so that it can display a wide variety of different signals on its outside. And depending upon what other immune cells come and receive these messages, the dendritic cell basically says, hey, look at what I found. I don't know if this interests you at all, right? But this is what I'm finding. In the case of a B cell, it's more specific, because a B cell doesn't just go around and taking everything in. A B cell has immunoglobulins on its surface. So in this case, IgM, the first type of immunoglobulin that a B cell would make, right? So the surface of this particular B cell would have many, many, many of these IgM molecules on its surface, and all of these IgMs would have the same antigen binding domains at the end of its arms, right? So this particular B cell knows how to make this shape of IgM and this shape uniquely. Right? So in this case, the green, the blue, the red proteins that were happily taken up by the dendritic cell, right? The B cell is more discriminatory, right? It only takes in the red one because it happens to be the right shape that its IgM binds to. And then the B cell, in a process of constantly recycling proteins on its surface, it pulls in lots of proteins from its surface, including the immunoglobulins that carry in the red protein here. It gets processed into these little red dots, which again are short little oligopeptides. And the B cell, just like the dendritic cell, right, loads these up on MHC2 and then displays this red dot, this short particular peptide on its surface. Right? So what does that mean? In this particular case, the very fact that the B cell is presenting this short peptide on its surface means that this short peptide is the thing that the B cell's immunoglobulin was designed to recognize. Right? So this is a pretty specific scenario compared to the top of the cartoon here, which is the dendritic cell just showing everything. So here you have a dendritic cell, right? So it is displaying, the red dot on MHC2 on its surface. Keep in mind, its whole surface would be covered with MHC2, displaying the green dots, the blue dots, the yellow dots, whatever, right? But let's just focus here on this particular case, this red dot. Right? So in this case, you have various helper T cells that are floating around. Each has a unique T cell receptor on its surface. Again, the whole surface of these T cells would be covered with their unique TCR, right? That's the one thing I always remind students, because it's really easy when you get caught up in these cartoons to think that somehow this purple cell has this humongous weird thing attached to it, right? No, right? The whole surface is covered with T cell receptor. But the point is the shape of this T cell receptor, or the green one or the red one, it's not the right shape, right, to bind to this red dot. And therefore, these T cells don't care. Okay, so that's sort of an unproductive interaction between the dendritic cell and the T cell. Oh, but in this case, this blue T cell over here, it has a T cell receptor that's the right shape to recognize the red dot in the context of that red dot being shown on MHC2. So this would be a productive interaction. So what happens is this helper T cell activates, right? It gets very excited. That's why it starts, I don't know what this is, it starts jiggling or something like that, right? Well, I think it's meant to be, it's supposed to send out different signals. And what is it looking for? It's hoping to find that B cell, the B cell that also specifically took in the red protein, because the B cell had the right IgM on its surface, right, to bring in the red protein. And then the B cell is presenting the red dot, the short peptide that came from that protein on its MHC2. So you have this helper T cell that's woken up, it pairs up with this B cell, and this helper T cell then tells this B cell, oh, you have the right shape Ig on your surface, right? You're important. You better go ahead and activate as well, right? So this B cell activates, it might make many copies of itself, and some of those B cells are then told by the helper T cell turn into a plasma cell, right? And make buckets, loads and loads of this Ig, but now make it, instead of the form that's stuck or anchored to the surface of the B cell, these would then be the released antibody forms, the soluble form of the immunoglobulin that can now spread out through the blood, right, hoping to detect more incidences or instances of this mysterious red protein that somehow might be coming from, let's say, a pathogen. Okay. So that is sort of the classic way that you think about T cells with the help of dendritic cells, showing them stuff, then working with B cells to produce antibodies to fight something like a pathogen, like a foreign invader, right, that's infecting your body. But in this class, what I want you to really think about is this mechanism of antigen-presenting cells showing things to effector cells like T cells, right, is how our immune system is constantly also surveying or monitoring our tissues for the evidence of cancer. So here, you can imagine that you would have something like a dendritic cell that has picked up on different sorts of funny sort of antigenic signals that are coming from maybe a cancer cell, right? Cancer cells are really sloppy, they're really messy, there's all kinds of weird mutant proteins that get released from them. This dendritic cell has picked up the red dot here. It is showing this to a T cell precursor. In this case, this would be a cytotoxic T cell, T sub C, right? And at the same time, it's showing it to this helper T cell, right? This T sub H. Right? So just like in the previous slide, the helper T cell will activate and it wants to help out other cells. In the previous slide, it was helping out a B cell, right? Letting the B cell know, hey, you should go ahead and turn into a plasma cell and make tons of antibodies. In this scenario, it's helping its cousin, the cytotoxic T cell. The cytotoxic T cell that also recognizes the same dot. And this helper cell tells this cytotoxic T cell, yeah, you're seeing this from the dendritic cell. I'm seeing it too. And I'm telling you right now, right, you need to take this seriously. You should go ahead and activate against this mysterious little peptide here. So the cytotoxic T cell matures and it becomes now, right, a mature cytotoxic T cell that's now floating around somewhere trying to find what cell in the body was actually showing this mysterious red peptide that it recognizes. And so here you have an antigen displaying target cell, right? So this might be a cancer cell, for example. Right? So on its surface, using MHC class 1 platforms, it's constantly advertising what's happening inside of it. Much of what's happening inside of it involves proteins that are still normal, everyday proteins, right? So maybe the brown dot here, the blue dot, and the green dot just represent common everyday little peptides from common everyday proteins, right? These are things that your immune system wouldn't really care about, right? But, right, because this is a transformed cancer cell, it is the source of the weird, mysterious mutant red protein. So it also displays the red dot on MHC1, right? And that's what the activated cytotoxic T cell, right, is looking for. So in this case, the activated cytotoxic T cell then will go ahead and send apoptotic messages to this target cell, telling it to go ahead and undergo apoptosis, right, and eliminate itself, because there's something wrong with it if it's going to be displaying these mysterious peptides. Right? So natural killer cells are another type of lymphocyte, and like T cells, they also induce apoptosis of targets. Right? The difference being that natural killer cells don't have specific T cell receptors on their surface that are very discriminatory and looking for like the red dot being presented by another cell. Right? Natural killer cells just have an array of different natural killer cell receptors on their surface that evolutionarily are very useful for detecting sort of strange signals being presented by other cells. So not surprisingly, we've evolved so that our natural killer cells have receptors on their surface that are very good at detecting types of strangely modified proteins that are indicative of cells that have transformed and started turning into cancer. So in this electron micrograph, you have a couple of natural killer cells that have synapsed or encountered for example, this big brown thing in the background is a breast cancer cell. If this were a normal breast cell, normal healthy breast cell, the natural killer cell might synapse, kind of check it out, but there'd be no stimulation of these receptors to actually get it to do anything. So the natural killer cell would just kind of detach, float off, and bump into the next cell, right? But in this case, because when it bumped into the breast cancer cell, some of the natural killer cell's receptors actually detected different signals that indicate this is a cancer cell. You can see now the natural killer cell has really spread out to really increase the surface area, synapsing with the breast cancer cell, and it's going to now send all kinds of pro-apoptotic messages. Here, down at the bottom, you have a natural killer cell synapsing or pressed up against a leukemia cell. Right? And so you can almost imagine, it's kind of fun to think that in between these two membranes that are pressed together really tightly, there's all kinds of signals going back and forth. Right, as proteins on the surface of the NK cell detect their ligand or the particular shaped funny molecule on the surface of the leukemia cell, right, that these receptors are designed to detect, and the natural killer cell sends messages to the leukemia cell after 60 minutes. Ooh, look at the fate of the leukemia cell, right? That's pretty intense, right? So this leukemia cell is undergoing apoptosis now, and it is just imploding. It's a very dramatic picture. It almost looks like the cell membrane is like collapsing inward, right? And all the guts of the leukemia cell are being digested and destroyed by caspase activity. Right? Cool series of images. All right. So when you have an immune cell pressed up against something like a transformed cell, and you get the proper activation of the immune cell, these represent external apoptotic signals. These are two examples here. The two main ways that a immune cell can externally tell a synapsed cell with it to undergo apoptosis. One of them involves the death receptor or FAS receptor on the surface of the cell. Right? So this bottom part is the FAS receptor. It's also called the death receptor. If it receives this FAS ligand or FAS-L that's being given off by the immune cell that's sort of detected that there's something wrong with the cell beneath it here, right? And it stimulates the FAS receptor. This FAS receptor then activates a bunch of pro-caspases and turns them into active caspases. Remember that these caspases are always present inside of the cell. These are proteolytic enzymes that would love to go ahead and start chopping up all these other proteins and other macromolecule components inside the cell. Right? But thank goodness, normally, right, they're held in a pro-caspase form because their peptide sequence, right, the way that they're wrapped up, oftentimes there's a part of the protein that covers up the catalytic domain. Right? But if that part of the protein gets cut off and the catalytic domain is then activated, right, you've now gone from a pro-caspase inactive form to an active caspase that now can go ahead and start chopping up other targets. Right? So the FAS receptor, the death receptor being activated, right, can activate pro-caspases that then activate sort of a downstream chain of different caspases that become executioner caspases, right? And these executioner caspases then go to work, right, chewing up all kinds of different proteins that make up the cell, things that make up, for example, components of the cytoplasm, like the cytoskeleton. On the other hand, cytotoxic cells, like the cytotoxic T cell that has joined up and synapsed with this particular target cell, right? If this cytotoxic T cell activates, it can release different types of cytotoxic cytokine messages, for example, granzymes, perforins, things like that. Those would then go ahead and activate other types of caspases that do things like activate or cleave protein intermediates that result in the mitochondria releasing the cytochrome C. Remember, we learned that the release of cytochrome C from where it's supposed to be held in the mitochondria into the cytoplasm, right, is the signal for apoptosis, because the cytochrome C binds up with these APAF1 proteins to form the spinning wheel of death, right? And that spinning wheel of death activates caspase-9, and then that caspase-9 will go ahead and activate a whole bunch of downstream execution or caspases that way. Right? So in both of these cases, right, these are external signals coming in from the outside. One that is very immediate, the FAS receptor, the death receptor, just going ahead and activating a whole bunch of caspases. The other involves things like caspases that activate and cause the release of cytochrome C from the mitochondria. Now, we did learn previously, when we first learned about or discussed apoptosis in this class, we had discussed apoptosis as being triggered in an internal or intrinsic way of triggering apoptosis, right? And that came not from external signals, but that came from things like p53 being activated, right? So if p53 detects that there's DNA damage, for example, right, p53 will want to activate and modulate different types of BCL family members, like BCL2, BAX, and BIM, that go ahead and open up the sort of floodgates of the mitochondria, releasing the cytochrome C, right? So in this case, though, what we're saying is, if for some reason this transforming cell was not successful in activating its own intrinsic p53-mediated, cytochrome C-mediated apoptotic signals, right? That's supposed to be what happens. It's supposed to be the cell detecting that it can't fix its problems, and so it does the right thing and hits the self-destruct button to take itself out of the picture, right? To spare the rest of the body cancer. Right? But if there are mutations that have gotten in the way of intrinsic apoptosis happening, right? So that's our first line of defense. Then our second line of defense is our immune system has to come, right, and actually tell the cell to undergo apoptosis. So how is it that cancer cells can get around this or even use this to their advantage? So many tumor cells, because of random mutations, can develop resistance to FAS-L signals. Something as simple as random mutations knocking out the production of normal death receptor on the surface of the transforming cell, right? Because as the transforming cell is getting more and more messed up, you would imagine, right, randomly, it's going to lose the ability to make certain types of critical cell membrane proteins, including things like, we already examined this. It might turn off the production of MHCs so that it no longer presents sort of incriminating evidence on its surface that it's actually a transformed cell. It may also lose the expression of death receptor on its surface, so they become resistant to the FAS-L signal. And a real kicker is, what if it no longer responds to FAS-L signal, but the FAS-L gene inside of this transformed cell that normally the cell shouldn't be making FAS-L ligand, because it's not an immune cell, let's pretend, right? But what if it gets mutated? So now it starts making tons of FAS-L. Now, right, it has a real advantage. It can make tons of death ligand, or big cloud of death ligand surrounding it, right? But it no longer responds itself to the death ligand. Right? So boy, this is a kind of a bad situation, right? So here are some melanoma cells. The nuclei are stained in blue, and in red, they've decided to stain for the presence of FAS-L protein, right? So this is really scary, right? So these melanoma cells are making tons and tons of FAS-L ligand now. And you can see that in this image here, all of these vesicles inside of these cells, right, are loaded up with FAS-L ligand. So that means they have sort of like a cloud of death ligand around them. So what happens when immune cells come and try to synapse with them, right? The immune cells are killed by the FAS-L ligand, because the immune cells are doused with so much FAS-L ligand and they still have intact death receptor, right? So they're told to die, right? So these melanoma cells have essentially built up like a shield or a force field around them. You can see this in these slides here. Yeah, sorry, these slides for some reason on the PowerPoint are all grainy. It didn't used to be like that, so maybe something happened to my PowerPoint file. But you can see them in the book. But even then, I think you can get an impression of what's going on here. Okay. So here are examples of tumor histology sections. So in brown, they are staining for the presence of Fas ligand. Right? So you can see these carcinoma cells are making a ton of Fas ligand. Right? And if you were to take that same tissue, instead of staining for Fas ligand, you want to see how many tumor infiltrating lymphocytes are actually succeeding in getting into the tumor to attack the tumor cells. You can see in red here, there are very few of these lymphocytes that are successfully getting in here. Why? Because they're being killed off by the Fas ligand that the tumor has learned how to make. Right? Over here, you see a situation where you have a tumor that has low levels of Fas ligand. So these tumor cells haven't yet learned how to make something like Fas. And sure enough, if you look at those cells, right, and stain for tumor infiltrating lymphocytes, you can see a lot more red. That means these immune cells are successfully getting in here and attacking the cancer cells. Another crazy twist to this is certain mutations in cancer cells might not only enable them to make lots of Fas ligand, but it may actually encourage the Fas ligand to be packaged up in vesicles that get released from the cell. And these vesicles, these little bubbles of death signals, right, start floating around in the blood. And so that's what these are here. So here you have a cell that's producing these vesicles filled with Fas ligands. This is a cancer cell. And these vesicles get into the blood, and that's what you see in the larger image. These are individual vesicles, little bubbles, lipid bubbles filled with death ligand that are now flowing through the blood, right? And as these crash into things like white blood cells, right, they fuse with the white blood cells, right, and they start killing off other immune cells in the system, right? How awful is that? Right? How crazy is that? So the evidence that melanoma cells, right, are very much still fending off. They have to resist or find ways of keeping lymphocytes from attacking them, is shown by this experiment here. So this is figure 15.32 in the book, where they describe a lab experiment involving mice. So in these mice, the blue bone marrow represents the fact that you have normal normal lymphocytes that are being produced with normal receptor for TGF beta on their surface. So TGF beta is a signal that inhibits or counteracts or tells lymphocytes to go ahead and die. Right? So you can see here, if these mouse lymphocytes, right, have normal TGF beta on them, in the experiment, what they've done is they've taken some melanoma cells, right, and actually implanted them into the tissue, into the bone marrow. And so these melanoma cells, they've learned to make or stumbled upon mutations that make a lot of the TGF beta signal, right? So that TGF beta signal gets bound up by the TGF beta receptor on these lymphocytes, and that causes the lymphocytes to be killed, right? And as a result, the lymphocytes don't make it to the melanoma cells, right? They leave the melanoma cells alone, and the melanoma cells then thrive and form a tumor. But in this experiment, they engineered these mice that have a double negative TGF beta receptor knockout. So in the green bone marrow right here, right, the TGF beta receptor that's made on these are a funny shape, right? That's what makes them a dominant negative genotype, right? So as a result, these green lymphocytes, now same experiment, right? You take these melanoma cells, you introduce them into the bone marrow, the melanoma cells make the TGF beta, but these lymphocytes no longer receive the TGF beta. So as a result, they are like impervious to this signal, and the melanoma cells and the green lymphocytes successfully get to the melanoma cells and kill them, right? So that you don't get a tumor. So this is a reminder that these random mutations that the melanoma cells come up with, right, they actually have to work to somehow counteract the immune system, right, allowing selective pressure for these subsets of carcinoma cells that are, in the tumor to be the ones that successfully can keep growing, while other subsets or subpopulations of the tumor cells can still get killed by the immune system. Another very clever way that cancer cells can learn to avoid or evade our immune system is by messing with the complement pathway. So complement proteins are a family of different proteins, there's like 30 different proteins that are floating around in your blood. And these proteins attach to the surface of cells such as pathogens, right? So whether you're talking about a bacterial cell, these complement proteins can attach to the surface. If you're talking about a virus particle, not really a cell, but a virus particle, right, these complement proteins can attach to the surface of a virus particle. And when they attach, they can do things like make it easier for other immune cells to be able to detect and destroy that cell. And importantly, these complement proteins can actually disrupt the cell membrane. So in this case, you have a series of complement proteins. Here, you don't have to really know this, but C6, C7, and C8 that are organized by this complement protein C5b. And they recruit this complement protein C9. And so several of these C9 proteins get recruited and they form a ring, and it basically pokes a hole in the membrane. So you can see here numerous, this is a cool image, right? Numerous of these complement attack complexes that are formed. And what happens is the cell can no longer control things like osmotic pressure, right? So water would come probably flowing into the cell uncontrolled, causing the cell to swell, and all the nutrients and proteins and things inside of the cell would leak out, right? So this could be very effective in, for example, targeting cells for destruction. Normally, the healthy cells of our body have proteins on their membrane that counteract this, right? This is why these complement proteins aren't always destroying your normal healthy cells. One of these is this protein known as CD59. Right, so CD59, you can see here on the right, it's this big ribbon structure that's being shown here, and it shows that it has like an anchor part of the peptide sequence, anchors it and keeps it on the cell membrane. CD59 is the right shape to bind and sequester or take away complement protein C9. So normally, the cells, healthy cells in your body, have a certain level of CD59 on them, so that if by mistake these types of membrane attack complexes start forming on one of your body's normal healthy cells, right, the CD59 on the surface can kind of take this apart before it causes any trouble. Right? However, what if you had a tumor cell, right, a tumor cell that should be actually attacked by the immune system? So if you have things like antibodies binding to the tumor cell, those antibodies should be really encouraging more and more complement proteins to come and try to destroy the tumor cell. Right? If the tumor cell just had normal levels of CD59 on its surface, it probably couldn't keep up with that, and it probably would get destroyed by your immune system that way. But what if one of the mutations that this particular tumor cell stumbled upon, right, is massive amplification or upregulation of CD59, right? So then the tumor cell might have way more CD59 on its surface than a normal cell, and it therefore is able to prevent the immune system from using complement proteins to destroy it. Okay. And this is an illustration that really highlights this. So these are cancer cells, right, mixed in with normal cells. And so what they've done is they've stained for the presence of CD59 on all of the cells in this sort of field of vision right here. Right? And you can see the threshold here is that the normal cells all just turn out to be black because they don't have enough CD59. In other words, the normal level of CD59 on their surface is not enough right, for the way that this experiment is calibrated to be able to see a strong CD59 signal, right? But that's certainly not the case for these carcinoma cells here, right? These cells have really stumbled upon mutations that dramatically upregulate the levels of CD59. So you can really see these cells glowing in this image, right? Because their surfaces are covered with the CD59. Okay. Okay, so yeah, so I think today's a shorter lecture, so that's the last slide for today. So let me go ahead and stop the share and stop the